Hello, and welcome to our series, What to Look For, where we talk about specific considerations for investigating foodborne illness outbreaks. In this video, we will discuss specific considerations for investigating Salmonella outbreaks. First, let's go over some of the basics of Salmonella. Salmonella is a bacterium that can infect humans and cause disease. Salmonella can fall into two major type classifications that also determine the type of infection they cause. One type is typhoidal salmonella, which is invasive and typically causes systemic infection in the body and a disease called typhoid fever. On the other hand, non-typhoidal salmonella typically causes infection of the gastrointestinal tract and a disease called salmonellosis. In the US, cases of typhoid fever are often associated with international travel, while cases of salmonellosis are often associated with consumption of contaminated food products, including various poultry products, eggs, fruits and vegetables, spices, chocolate, flour, and many other type of food products. Given its strong association with the U.S. food supply, in this presentation, we are going to specifically focus on considerations for investigating outbreaks linked to non-typhoidal salmonella. Non-typhoidal salmonella, which we will refer to as Salmonella for the remainder of this presentation, causes approximately 1.35 million cases of salmonellosis in the U.S. each year. Common symptoms of salmonellosis include diarrhea, fever, abdominal cramps, and vomiting. Generally, Salmonella has an incubation period ranging from 6 to 48 hours, and people with salmonellosis typically experience symptoms of foodborne illness between 4 to 7 days. Common local establishments where Salmonella outbreaks can occur include restaurants and other eateries, long-term care facilities, schools, and penitentiary facilities, just to name a few. When investigating Salmonella outbreaks linked to local establishments, there are a variety of areas where environmental health investigators may need to focus their investigation efforts. These include evaluating whether improper cooking practices could have contributed to the outbreak, evaluating if cross-contamination of Salmonella during food handling or storage could have contributed to the outbreak, and evaluating whether infected food handlers may have been the source of the outbreak. Additionally, environmental health investigators may also need to take steps to evaluate whether environmental sources or contaminated food products or ingredients may have been the source of the outbreak. First, let's discuss how to evaluate whether improper food preparation practices, such as cooking, could have contributed to the outbreak. Retail and food service establishments that prepare food need to make sure raw foods of animal sources like poultry, meat, or egg products are cooked to proper temperature and time to ensure that any salmonella that is potentially present on these raw ingredients is inactivated or killed. Remember that, although the poultry industry is doing everything to eliminate salmonella from raw foods like poultry and eggs, there is always a chance that salmonella could be present on these foods before cooking. To evaluate whether improper cooking practices could have contributed to a salmonella outbreak, environmental health investigators should identify whether any cooking practices used in the establishment were not appropriate to ensure an activation of salmonella. This process should include reviewing relevant records in the establishment, such as 1. Standard operating procedures, such as SOPs or recipes, and 2. Documents where monitoring and verification activities are recorded. By reviewing the SOPs, the investigator is making sure cook procedures are appropriate, and by reviewing the monitoring and verification records, the investigator is making sure these cook procedures are consistently being followed. In addition, investigators can also review other documents that are relevant. For example, logs that show how often thermometers used to monitor the cook step are calibrated and what calibration records say about the accuracy of the thermometer. To supplement this review of records, investigators should also conduct interviews with both management and food handlers at the establishment and ask questions about 1. How SOPs for cook steps are enforced, 2. Any potential inconsistencies in SOPs and monitoring or verification records, as well as other questions about 3. Food thermometer calibration and maintenance. Whenever possible, investigators should observe or ask food handlers to demonstrate the cooking procedures. Next, we will discuss how to evaluate if cross-contamination of Salmonella during food handling or storage could have contributed to the outbreak. Salmonella can be spread from contaminated food products to non-contaminated food products if food handlers are not washing their hands or changing gloves between handling foods like raw poultry or meat and handling ready-to-eat foods such as fresh fruits and vegetables. Also, not properly cleaning and sanitizing equipment and utensils between handling different types of food can also potentially facilitate cross-contamination of Salmonella. Additionally, poor separation and storing foods like raw poultry or eggs above ready-to-eat foods in refrigeration units can, in some instances, facilitate cross-contamination of Salmonella, as the juices from these raw ingredients, at higher risk for being contaminated with Salmonella, can drip down onto the ready-to-eat foods. To identify whether food handling or storage practices could have facilitated cross-contamination of Salmonella, environmental health investigators should evaluate the practices used to prepare and store food products in the establishments during site visits. This can be done during observing how food is routinely handled in the establishment. During these observations, one key thing to take note of is the accessibility of hand-washing sinks and gloves. For example, are hand-washing sinks properly stocked with soap and paper towels? Are gloves available and easily accessible to food handlers? How frequently are employees washing their hands or changing their gloves? Are food handlers changing gloves between handling raw ingredients and handling ready-to-eat ingredients? Additionally, pay attention to how equipment and utensils are segregated. Are food handlers cleaning and sanitizing equipment and utensils between handling raw ingredients and ready-to-eat products? Or is there a color coding system in place? For example, are different color cutting boards used to handle raw ingredients and ready-to-eat food products? Also, note how food is stored in refrigeration units. Are any raw ingredients stored above ready-to-eat food products, such as fruits and vegetables? Are all raw ingredients stored in leak-proof containers? Such observations are crucial, as failure to properly segregate raw ingredients from ready-to-eat foods during handling or storage can increase risk of Salmonella outbreaks. For example, in a 2017 Salmonella outbreak associated with a restaurant in Colorado, environmental health investigators conducted a site visit to investigate the outbreak, where they observed multiple food safety violations, including 1. Raw chicken in a non-leak-proof container being stored on top of a box of ready-to-eat tortillas in the walk-in refrigerator, as well as 2. A food handler who failed to wash their hands or change their gloves between cracking raw shelled eggs and handling ready-to-eat tortillas and cheese. Additionally, investigators can also conduct interviews with both management and food handlers about these routine food handling and storage practices. Important questions to ask during these interviews include how is hand washing enforced in the establishment, as well as are food handlers encouraged to wash their hands or change gloves between food handling activities? Are there instances in which food handlers are advised against using hand washing sinks or changing gloves due to scarcity of supplies? Also, it is important to ask food handlers on how they have been trained to properly segregate raw food ingredients from ready-to-eat products and how these segregation practices are enforced by management. Next, let's move on to discuss how to evaluate whether infected food handlers may have been the source of the outbreak. This can be done by reviewing relevant documents, either during or prior to site visits, such as any available employee call-out records, sick leave documents, as well as other relevant documentation on recent illness reported by employees of the establishment. In addition to reviewing records, environmental health investigators should also conduct interviews with both management and food handlers within the establishments during investigational site visits. Some examples of important questions to ask in these interviews include if and when any food handlers or other employees started showing symptoms of foodborne illness, whether there is an issue with short staffing or high rates of turnover, and how sick leave policies are implemented and enforced. Gathering information on how sick leave policies are regularly enforced is especially important because if written sick leave policies are not appropriately enforced, the establishment could be at an increased risk for Salmonella outbreaks. If one or more cases of foodborne illness are identified among food handlers or other employees based on record review and interviews, it is important to identify the exact dates in which these individuals reported showing symptoms, when they attended work, and how these dates correlate with estimated exposure dates for people affected by the outbreak. This information can help investigators identify whether the infected food handlers could potentially have been associated with the outbreak. For example, in this scenario epidemic curve of a Salmonella outbreak, food handler A first reported showing mild symptoms of foodborne illness at work on both April 26th and 27th, but did not call out sick from work until 28th, when their symptoms became more severe. As we know that Salmonella has a typical incubation period of 6 to 48 hours, in which individuals may still be able to shed the Salmonella without showing symptoms, mapping when food handler A started showing symptoms and when they stopped attending work to this scenario epidemic curve provides evidence to support that this food handler may have been a potential source of this Salmonella outbreak. Note, in a handful of instances, food handlers or other employees might have asymptomatic Salmonella infections, meaning that they are harboring the bacteria but are not showing symptoms of foodborne illness. Asymptomatic Salmonella carriers can potentially shed the bacteria for long periods of time, which can increase their risk of potentially contaminating food products. For example, in a 2002 Salmonella outbreak associated with salsa at a hotel restaurant in Texas, hotel management requested that all food and beverage staff submit stool samples for Salmonella testing to support the outbreak investigation. From this testing, it was revealed that a sous chef responsible for preparing salsa product in the restaurant tested positive for the outbreak strain. Interestingly, however, this sous chef did not report showing symptoms of salmonellosis either before or during the outbreak period. Next, let's discuss how environmental health investigators can go about evaluating whether environmental sources in the establishment may have been the source of the outbreak. Salmonella can survive well in various food production and preparation environments. This includes both wet production environments such as retail delis as well as dry environments such as bakeries. And Salmonella can live in various types of equipment or facility structures, including in floors and drains, as well as in metal-to-metal junctures in equipments or tables, food utensils, and food mixers or bowls. Salmonella present in any of these areas in the food production or preparation environment can potentially contaminate food products, increasing the risk of foodborne illness outbreaks. For example, in a 2023 Salmonella outbreak associated with a restaurant in Maryland, environmental health investigators collected several environmental samples from the establishment during the outbreak investigation. A handful of the samples collected from specific sites, including equipment wheels and floor drains, yielded the outbreak strain. To evaluate whether environmental sources in an establishment could have been the source of the outbreak, environmental health investigators may want to select environmental samples from the establishment and test them for the presence of Salmonella. Some key things to consider when collecting environmental samples include making sure that the right tools are used to collect samples. For example, using sponge kits with sterile gloves or sponge on sticks are typically preferable to using cotton swabs, as they can cover a wider surface area and capture more environmental microbes. Additionally, in some cases, investigators may have to disassemble certain types of equipment, such as this deli slicer, in order to access hard-to-reach sites inside the equipment that could potentially be harboring Salmonella. This may require additional support from maintenance personnel at the establishment to properly break down the equipment. Finally, let's discuss how environmental health investigators can go about evaluating whether contaminated products or ingredients may have been the source of the outbreak. In certain instances, food ingredients may be contaminated with Salmonella before they are purchased or obtained by an establishment. In particular, when investigating an outbreak associated with a single local establishment, these contaminated ingredients are often sourced from local farms or small-scale local producers. Examples of some of these food products or ingredients that might be at high risk for contamination with Salmonella before they arrive at the establishment include eggs, particularly if they are sourced from local farms, as well as various vegetables, herbs, and fruit. To evaluate whether contaminated food products or ingredients may have been the source of the outbreak, environmental health investigators may want to select food or ingredient samples from the establishment and test them for the presence of Salmonella. Some key things to consider when collecting food product samples include making sure that sterile or sanitized tools or utensils are used to collect samples. For example, if you are using utensils, such as knives or tongs, from the establishment, be sure to clean and sanitize them before collecting food product samples. Additionally, it is important to ensure that all collected samples are transported to the lab appropriately. More specifically, samples should be transported under similar temperature conditions in which they were collected. For example, if samples were collected at 40 degrees Fahrenheit, then they should be transported to the lab at or around 40 degrees Fahrenheit. In summary, in this presentation, we have covered some of the specific characteristics of Salmonella, as well as 5 key areas that are relevant for environmental health investigators to consider when investigating Salmonella outbreaks linked to local establishments. In addition to Salmonella, check out our other videos in this series that cover key considerations for investigating foodborne illness outbreaks linked to other foodborne pathogens.